about some of the work that we are doing at Essex, where we're looking at how crops and climate influence crop productivity and how plants can cope with that climate change. And we're just going to tell you a little bit about some of our research and how that contributes towards this topic. Okay, so I'm Tracy Lawson. I'm a professor in plant physiology at the School of Life Sciences at the University of Essex. And just for any of you that have not been there, this is our campus, and we're located in this building down here. You can usually see that because there's a greenhouse on the roof that you can see. Usually has some brightly coloured LEDs in there. And this is our new STEM teaching centre. So plants are really, really important for climate and for our planet. We would not be here without them. It's plant photosynthesis that actually put the oxygen into our environment that allowed us to develop and evolve. But also, you can see here, this is the heartbeat of plant productivity. And what you're seeing here is that as we go through the different of the different months here, you can see that the green is the productive areas, that goes red for productive areas, and this is gross primary production, how much of the plants are taking in CO2 out of the atmosphere. And as it changes through the different months, we can see that the northern hemisphere in the summer has really high productivity, really drawn down the CO2 out of the atmosphere, and then the same in the southern hemisphere in the winter and less in the north. So really plants are the heartbeat to our planet and really important in terms of CO2 uptake and actually sequestering carbon dioxide from the environment. And if we look at the environment and the biomass of the planet and who's on the planet, this is the animal kingdom here that you can see. And just to give us a bit of an idea, this is humans. We evolved in a relatively short amount of time ago, but we are less than one sort of bar of chocolate here, chunk of chocolate in terms of the biomass of the planet. So we represent about 0.06 gigatons of carbon on the planet. We do quite a lot to the planet, having been only a small biomass of the planet, and we can see that there's the other animals in this part of the global biomass of the Earth. However, when we put that into perspective, just the animals alone, and the humans being a little chunk of this animal kingdom. This is the animal kingdom. There's various different other kingdoms, the fungi, the archaea that you might have heard about this morning. This is the plant kingdom, by far the biggest biomass on our planet. And plants make up over 82% of the biomass of Earth, more than 320,000 different species of plants, and really, really key to our global climate and productivity of the planet. So humans make up about 0.01% of the Earth's biomass, 0.06 gigatons of carbon. Plants make up 82.4%, and 450 gigatons of carbon are in plants. So really, really important for our carbon cycle, as well as our water cycle as well. So why are plants important? Why do we do a lot of research? It's a big research group for us at Essex. Well, as I mentioned, access to oxygen. Without plants, we wouldn't have that oxygen. They're here to oxygenate that planet, take in CO2 and release oxygen as part of that. But most importantly, most of our research focuses on the fact that they provide food for humans and animals. Everything we eat, whether you're eating your beef burger and say, oh, well, I don't like veg, your beef burger came from a cow that was eating grass or grain that was from a crop. Everything starts, and primary productivity starts with plants. Of course, they provide us fuel, not just fuel that we can burn now, like wood, etc., but coal was the plants that lived 250 million years ago and formed those first coal seams. And of course, materials. We get huge amounts of materials from plants. And it's amazing how much plant products are in various different products that you buy. Even toothpaste has some components of plants in it. Soaps, threads, papers, loads of different fibres that come from these plants. So what we research at Essex really is focusing on increasing plant productivity and yield and trying to make plants more resilient to changes in climate. And this is because we know that we need to increase food production in the next 50 years. We virtually have to double food production in the next 50 years, by the year 2050. That's not very far away. And of course we need to do that in the face of climate change. We know that CO2 is rising. Because of that, we know we've got the greenhouse effect and temperatures are rising. And then weather patterns are becoming more sporadic and erratic. So we have massive floods, then we have droughts, then we have a flood, then we have a drought. And so plants have to cope with all of that, and we need them to do that. Not with more land. We don't have more land because we've got increasing global populations. We have to do that with better crops. And that's what our research focuses on. So the major factors that mean that we need to improve these crop yields is, the biggest one, as I've just mentioned, is population growth. We are increasing at a rate that we've never increased before, and I'll show you a graph of how the population is changing in a moment. We've got shifting diets, so there's a lot of Western, countries moving to more Western diets that includes much more meat-based products. Makes it much more grain is required to make meat-based products than if you're eating a vegetarian-based diet. Increased urbanisation. With that population growing, we'll have more and more houses, and more urbanisation, less land. So we have to increase this food production with less land and less resources. And of course, unsustainable land use. We know that soil health is a real issue. We know that water is becoming an issue, nutrients, etc., pesticides, everything are all going to influence crop productivity. And then on top of that, we've got the changing climate that I've already just mentioned to you. So if we look at population size, this is the biggest need to increase productivity. So if we look, this is a graph of the past sort of changes in the population over the last 12,000 years. So we are actually way past that now. We've stopped there at 2019. But just to put this into perspective, in 1975, when I was about 4 or 5 years old, this is me with my twin sister in a tractor. I grew up on a farm, so I've always been interested in crops and farming. There was about 4 billion people on the planet. By the time I went to university, or started studying at university a few years later, there was another 1 billion people on the planet. And then by the time I went to Essex in 1999, there was another 1 billion people on the planet. And we're now sitting at about 7.9 billion people on the planet at the moment. And that's predicted to stabilise at the end of this century at around 10.5 billion people on the planet. So if we think about the food that we needed in the 60s relative to what we need by the end of this century, there's huge, huge differences in that food that we need. So as I say, we need to really double food production by the year 2050. By the end of the century, we have to produce more food than we have ever produced in time before. So these are huge challenges that's facing the agricultural sector at the moment. And we have to do that in the face of climate change and with no extra land. So just to give you an idea of the requirements that we need, these are some of the key crops that we focus research on globally, and as well as the work at Essex. And if we look at what's happened to yield, so this is the actual yield increases in these major crops of maize, rice, wheat and soya. So these little lines are how yield has increased with time due to various agronomic practices, breeding practices, etc. This is the line, it's the trajectory line that if we carry on as normal is where we'll reach. And the dotted line here, this dashed line, is what we actually need to do. And so you can see that as we go further and further towards 2050, our gap in what we can do and what we need to do is increasing. So we need a step change in how we're going to try and produce food in the future. So do we have enough plants for food? And at the moment, no. We don't have sufficient production, and definitely not for the future. We can see these gaps widening. And of course, there's main drivers of this as well. As I mentioned, we've got this need for increased food production with growing populations. But on top of that, we've got these changes in the climate. And so we've got an increase in atmospheric carbon dioxide concentration. And again, this graph here shows you time on the x-axis. So we're past this now, and global temperature changes and CO2 concentrations. So the line here shows you CO2 concentration, and the bars show you the changes in global temperature. And as we know, we've been pumping CO2 into the atmosphere. So when I was a student, we used to set our machines for atmospheric CO2 concentration at around about 352 ppm. I was told as a student CO2 concentrations were rising by 1 ppm a year. That's 1 part per million a year. We now set our machines at about 419 parts per million. And so I am not over 70 years old. I hope you believe that as well. So we're now rising CO2 concentration about 2.3 parts per million a year. So we learned about climate change again when I was a student, and it said, oh, this is the scenario we need to do. This is our worst case scenario. We actually superseded our worst case scenario and went even worse than that 20 years ago. So now that CO2 concentration is rising, we're not stopping that. And of course now it's trying to mitigate the effects of the temperature increases on that. If you increase the temperature, the plants need more water. We're going to have less water on the planet, more sporadic rainfall on the planet. But also, photosynthesis is a biochemical reaction. Enzymes are involved in that. Temperature will affect those processes. So things like wheat are very temperature sensitive, and Amanda will tell you a little bit about some of the work that we're doing, looking at temperature sensitivities and ways to maybe mitigate against that. So these are two main drivers, again, along with our population increase, that means that we need to increase productivity. So a lot of my research focuses on photosynthesis and water use in plants. So just thought we'd look at a scenario here. If you've just had your lunch, maybe you had steak sandwich and a beer, for example. So how much water do you think is needed to make this lunch? We can calculate this. Let's work it out. Okay, so we take a bread, an average slice. This is not quite a slice of bread, but bread was easy to find. So bread takes an average of 1,827 litres per kilogram to produce a loaf of bread. An average slice is about 800 grams. It's a decent slice here of bread. So we need, an average loaf about 800 grams. That's about 1461 litres. So if we take 16 slices, 91 per slice. So 91 litres per slice. So we've now got 182 litres for our bread alone of that water. As I mentioned before, beef is going to be much more, trying to produce protein requires much more grain, much more water. So beef, 15,400 litres per kilogram of water is needed for beef. It's because you need to make the grass, get the cattle feed, grains, etc. So animal food being the main user of water for some of our food production. We've got a decent steak here, 18 ounces, 3,492 litres of water for that steak. And then beer. Again, 1,420 litres per kilogram, so a pint's around about 860 litres. So actually, our 1 pint of beer and 1 steak sandwich took 4,480 litres of water to produce. Okay, so these are huge numbers to produce your pint of beer and your steak sandwiches. We know that rainfall patterns are changing. We now need to find crops that's more water use efficient, and that's a key part of research in my lab. And so what we look at, and if any of you've been into the Planet Positive tent, we've got some thermal cameras and some stomatal pictures, etc. there. We look at the stomata on the leaf surface, and these are the small pores. Stomata means mouth in Greek. These are small pores on the leaf surface, microscopic, around about 20-30 microns in size. And these open and close in different environmental conditions. So the environment's very dynamic, so they tend to open with light, for example, close when it gets a bit duller. And they open and they allow the carbon dioxide in for photosynthesis, but as a consequence of being open, they also allow water out. Water leaves the plant much easier than the CO2 goes in. So we have an order of magnitude more water is lost than CO2 goes into the plant. That water loss is really important because, again, if you come across the Planet Positive tent, we can see that as the plant loses water, it's like us, we cool down, and that cooling can help those enzymatic processes that I mentioned before. But also it means that the plants are using a huge amount of water. So some of our research is trying to look at some of these stomatal behaviours and trying to alter them to try and speed up how quickly the stomata can open and close. If we can find plants that have faster stomatal opening, because they're a bit slow and a bit sluggish, which means that if they take a long time to open, they limit CO2 for photosynthesis, and that means they decrease productivity. Whereas if they take a long time to close, they can limit, they lose much more water than CO2. So try and find fast, speedy stomata. And so one way that we look at these is by using thermal imaging, that we've got some systems set up next door that you can see it. And basically, if you have the stomata open a lot, they will lose water and therefore cool the leaf down, and we can see the differences in these plants. So you've got an example here, we've got open stomata, losing lots of water, cooling down, and therefore they have a much lower temperature than this plant here, where the stomata are closed and a much higher temperature. So that gives us a great screening tool to see if we can then find plants that's more water use efficient. And it's really important to remember that the measure of how open those stomata are, we call stomatal conductance, is related to grain yield. The more open they are, the more CO2 you get in for photosynthesis, but also the more you cool the leaf and keep it happy for those photosynthetic processes. So it's a real balancing act of trying to make sure that they operate so you keep photosynthesis but not lose too much water. We don't want to close them. If we close them, we'll have no productivity. So it's really trying to balance the behaviour of those stomata. Again, we can a number of different ways we can do this. I talked about the speed of stomata responses. We also have research where we're trying to alter the number on the surface, and there's different genes that's related to how the number of stomata are on the leaf surface. And again, you can see this is an example of work from somebody else's lab where you can change the dots here represent the stomata, and the more stomata you have, the cooler the plant is, the more water it's losing, the more evaporative cooling that plant actually has. And we've got thermography also allows us to look at dynamic images, and again we can see some of this in the tent next door. You can see that as the light changes, we can see changes in those thermal patterns. That gives us an idea of what the stomata are doing, how quickly they are responding and changing to the different environmental cues. Okay, so I had to put a graph in here to show some of the work that we've been doing. We sort of do loads of these graphs where we look at the changes in these stomatal kinetics. This is some of our research that we've been done, but we've really simply, this looks really complicated where we're looking at the changes in that stomatal behaviour, how open they are, but really what we want to do is just time how long it takes them to open, and that's the simplest way we can do this with some of our kit. We have lots and lots of different stomatal responses depending on different species of plants. So you can see here we've got stomatal conductance in this black line, and then photosynthesis in the red line. We can see that these are really well coupled together. Whereas we can see for these plants we've got slow stomatal behaviour, and that limits photosynthesis. And these ones, again, very, very different. So there's lots and lots of different crops have much different stomatal responses and photosynthetic responses, and we've been looking at some of those. And basically what those tell us is that the slow stomata, on average, limit photosynthesis and crop productivity by around about 10%. So if we can speed those up, we can maybe improve yield as well as improve water use within the plants. And we're also looking at different mutants, so we know that there's mutant plants available. These are natural plants that have mutations in their genome, and this is just an example of stomatal close and how quickly the plants close. And we've got two different mutant plants here. So this is our wild type plant, has an average closing rate of that decrease in that graph. We've got one that's much, much slower, and then one that's much, much faster. So if we can take the knowledge of this gene and transfer that into different plants, whether that's a transgenic approach or a natural approach, by knocking that gene out, we can maybe get faster, speedier stomata and try and improve water use in our plants. And we're also looking at various different aspects of this red and blue light pathways in stomata, and blue light open stomata much, much more than they're needed, and that's another part of our research where we're trying to limit some of that. And if anybody's interested in that, I'm more than happy to talk to you in the tent about some of that research. Okay, so that's a whistle-stop tour of the research in my lab. This is my team that do this research, and just acknowledging that they actually do all the research. I just tell you about it, but these are the guys that's doing it on the front line. So if you've got any questions, feel free to ask.
The lunch you did, the pizza lunch?
Yes.
Would that be if it's a vegetarian? I'm a vegetarian.
Yeah, so...
What would that be if it's vegetarian?
Yes, much, much less. So, as I said before, yeah, meat is the massive driver of that water use. So if we think of that as being 3,500 litres of that, taking that off the main one, you're more than, well, a third of what you would do, a half to a third of what you would do if you substitute that for a vegetable in there.
So vegetable would be 2/3 less?
Most likely, depending on what you put in there, but yes, so the bread's a component, the beer's a component, the meat is the biggest component. So if you substitute that, and that's a massive reason why we're changing the amount of crops that we need, is because our diets, a lot of diets are shifting towards Western diets. In developing countries, they're shifting towards Western diets, and that's putting even more pressure on our grain requirements.
Just another question, you were talking about the stomata. Is that genetically modified?
So there's two approaches that we are looking at. So the density ones that I showed you was a genetic modification from other people's labs. We are exploring that as well, but we're also looking for the natural variation. And in the genetic approach allows us to find the target genes, it allows us to understand those genes, which then gives us the opportunity to find the non-transgenic approach. If we know how genes are regulated, you can find mutants in regulators that alter the gene that you want, but you have to have that knowledge before you can even start going down that. So we use transgenics as also a discovery tool and a development tool for our research.
Is there a grain that's more, better, like is rice better than wheat, for instance?
For water?
Yeah, with stomata.
So the one picture that I showed you, it had very, very fast kinetics, and then rice is really fast stomata.
So that's a good one.
So it's a good one. Wheat is really quite fast as well, but actually I sort of skipped over it because I felt it was getting a bit too sciencey. But the blue light component that I was talking to you about is that when we we call a red light just general light. We're just calling it red because it's not blue. When we put that on and the stomata open, so this is our stomata opening, they open and they're open enough to get that CO2 in for photosynthesis, losing a bit of water, but that's so optimal. If you put a tiny bit of blue light on, like 10 micromoles, which is this full sunlight's 2,000 micromoles, so put 10 micromoles on, very small, hardly enough to drive photosynthesis. That's what you can do to stomata. They open much more, but don't increase photosynthesis. So I am doing research where I'm trying to knock this out, and I'm doing that through natural populations. And doing that with John Innes Centre, where we're trying to knock out the blue light response and knock out this bit of that stomata response and just leave that. Then they'll photosynthesize the same, but lose less water. And that's the plan with this. I just thought it was probably getting a bit too much science.
Is there any other questions or before?
Excuse me, one question occurs to me. The amount of litres to make one sandwich in one beer seems so huge, it's almost unbelievable. Surely it must be recoverable or converted or changed. You don't lose all that water, do you, necessarily?
No, that's right. I mean, there's parts of that are recycled, but that's in terms of the crop actually going through that. So of course that's also going into the atmosphere and feeding back around in the cycles, etc. And so changes in what we grow and where we grow it and how that grows also changes our cloud condensation nuclei and the rainfall patterns across the globe as well. So yes, that's actually just what's going through the plant as a ballpark figure of water use. It's really to emphasise that it's much, much more than you'd think actually that's needed for that plant to grow, even though you capture some of it back. Yes.
That's good. Don't feel so bad now, thank you.
Did you have some beer? Okay, so I'll hand over to Amanda, who's going to tell you a little bit about the second part of our talk, a little bit more about some of our research at Essex as well.
Great, thank you. Oh, great. Thanks very much. It's nice to see you guys in here with us. Oh, the wrong way. There we go. On a day when we maybe wouldn't be thinking of the heat, unlike a day like yesterday. But the real question that's driving what I'm interested in scientifically, why you can tell I've come to the UK to do this work, obviously not from here, is really asking how can we increase crop yields in a changing climate. And so I remember sitting in a room, feel free to come in if you guys would like, I remember sitting in a room hearing lots of the projections that Tracy just walked us through about what the climate might look like at mid-century and how many mouths we will have to feed at mid-century. And I remember I was quite a bit younger and I was doing the math, and I realized that that is a career's worth of time for me in which to tackle this problem. And so it's been very much the driving force in the questions I've been asking. How are we going to be able to do this? And I think some of the questions that came up at the end of our last talk were a perfect introduction to this, because I think there are two main approaches we can use here. We can use conventional breeding techniques, right? And so anybody with a garden will be very familiar with some of the seed saving and crossing involved in conventional breeding techniques. Or we can use biotech approaches. And what I'm going to do today is walk you guys through. There are tons of seats up front if you guys want to rest your legs. But I'm going to walk you through a story of some of the work I've done that uses potentially both of these approaches. Okay? And so of course with conventional breeding, well, logistically, that's quite easy for us to fathom, right? So we have a trait that we want, a bigger plant with more tomatoes. We have a plant that maybe has something else, good disease resistance, speedier tomato perhaps. And so we can cross these, and over successive generations, we can start to find offspring that have both of the traits that we want, right? And so if we had food today, we can thank generations of plant breeders, because that is how the tomato came to our lunch. That is how we had the bread. That is how, for those of us lucky enough to have already had our first pint today, we've enjoyed our pint, right? So generations and generations of plant breeders have produced varieties of food that we need. And one of the things that really has been spawning interest, or like spurring plant scientists like me and like Tracy along, is that that's a really slow process. And so with each generation, you have to find the best performers, you cross those out again, you back cross them into something else, and it takes time. And unfortunately for us, time is one of the things we're up against, right? And so a few years ago, one of our colleagues published a paper really addressing this issue. And you know, they titled it quite dramatically, One Crop Breeding Cycle from Starvation. And I think, you know, that is dramatic, but it really puts a play on the time that we're looking at here. And the thing that they were really trying to point out is that a traditional crop breeding cycle takes about 30 to 50 years. And as we heard, we have major global challenges facing us in the next 25 to 30. All right? And so we have one chance to do it at this point by traditional breeding cycles only. And so another tool in the plant breeding kit to know which techniques we should be putting into these pipelines to have resources ready if we come to need it are biotechnological approaches. And so they test approaches, particularly that evolution can't solve. Plants, in my opinion, are amazing. Plants do so much for us, right? If we had a breath of air today, if we had something to eat, we can thank a plant. That did that for us. Feel free to come in if you would like. That did that for us. And so there's tons of things that plants do really well, but there are some problems even they can't overcome, okay? And so we are able to now test these solutions out using biotech approaches to see if they are. And so of course I wouldn't be a scientist if I didn't have an equation to explain even the most simple concept to you guys, right? And here I am with my fancy equation up here. But this is basically the yield potential equation. So for plant scientists, we spend a lot of time thinking of this, and this is basically telling us that the yield, the theoretical maximum yield from a crop, is the product of the amount of sun it's going to get, how well it can intercept that and use its leaves to intercept it, how well it can convert it from sunlight into carbohydrates, photosynthesis, and how well it can partition it into the grain. And if we actually take a look at how plants are doing this, what we find is that most of these are at its theoretical maximum, but photosynthesis is not. So for many evolutionary reasons, but one is my favorite that I'll talk about, plants have some fatal flaws. And so photosynthesis is not nearly as efficient as it should be. And the big reason why is that photosynthesis is a victim of its own success. And so it's generated this world that makes life possible for us. Thanks photosynthesis. And it's done that because it has this one enzyme, so this one little piece of molecular machinery in every leaf on the planet. Rubisco, my little buddy, I've been spending a lot of time thinking about this, has fixed carbon dioxide, which is a really tough job. So this enzyme in plant leaves can grab a molecule of gas, fuse it to a sugar, and then make all carbon-based life on Earth possible. So every atom of carbon in our bodies pass through this enzyme in some leaf on its way to our plate, on its way to us. All right? And that's not an easy job. And so it has one little problem. And that's like most of us, you can do a job well, you can do a job fast, but you can rarely do both. And so Rubisco makes one little problem in that about a quarter of the time, it grabs an oxygen instead. It's kind of confused, it's kind of sloppy. These two things look really similar, the gases look similar. Who hasn't made a mistake, right? And for a long time for Rubisco, it didn't matter at all, because oxygen evolved when there was not, or photosynthesis evolved when CO2 was high and oxygen levels were low. And the oxygen in the atmosphere that we enjoy breathing to survive came from photosynthesis. And so as oxygen photosynthesis became prevalent, as plants colonized the land, we see a problem because it's starting to fix a lot more oxygen. And we can actually model that out. And so if we model that out, this is looking at sort of the American bread belt. Bear with me, we've got to update some of these figures for European audiences. But if we look at the American bread belt under current conditions, okay, what I want you to see is just this swath of color, which might not be easy to see here, but we lose about 30% of our agricultural crop productivity, our maximum productivity, in crops like wheat, in soybean, in rapeseed, just due to this one mistake. So imagine if we made a mistake and we cost 148 trillion calories annually. That's what happens to my poor buddy Rubisco. And the real problem is that even as CO2 levels increase, temperature is increasing too. We're not getting one without the other. There are seats if you guys want to come in and take a load off, but we're not getting one without the other. And so even as CO2 increases, we're still seeing these crop decreases. And so how can we solve this problem? Nature has done it. There is a nature-based solution to this. Nature has solved this little rubisco problem by evolving a better crop. And so in crops like maize or sugarcane, these really hyper-productive crops, nature has put in a CO2 pumping mechanism. Okay, and so if you look at these leaves, they operate differently at a cellular level. They've concentrated CO2 around poor sloppy rubisco, so it doesn't feel the cost of that oxygen mistake. Okay? But, you know, we can do that. There are groups out there who are looking to re-engineer this nature-based solution, but we also asked, you know, is there a better way? So this is just the complicated pathway on the screen in front of us of what happens when rubisco makes that fateful mistake. And so it takes a lot of energy. It moves the compound that it makes instead of what we want into 3 different subcellular locations to try to get back to what it needs. And so I say it's akin to making a mistake in a recipe, and then instead of, you know, being able to just fix it while you're cooking, you have to go out to the shed and get something, and then you maybe have to pop by your garage or another room in the house to fix something, and then you can put your cake in the oven, right? It's clearly taking longer. It's clearly taking more energy. And so we tried to use a biotech approach, so a GM approach, to engineer a pathway that could contain everything to the chloroplast. And so instead of making this mistake and moving through, can we have rubisco make its mistake? Can we keep it all in the same location? And we of course used genetic engineering to do this, because this is a pathway that didn't exist in nature. But it was really important to see if we could see a benefit, because as Tracy said, first you have to demonstrate the benefit so that you can go looking for nature-based solutions. Okay? So we took our genes of interest from algae and a pumpkin. We isolate them using some pretty routine molecular biology methods. We put them in our plant DNA, and then once it's introduced, it functions like any other gene. You can cross it, you can collect it, you can grow it under very strict controlled conditions. And when we did that, what we found was that by fixing rubisco's mistake, we got plants that were about 30% larger. So this is our model crop. Tobacco forms a leafy canopy, produces a lot of seeds. You can get it grown within a year. So really important for testing out novel solutions quickly. We also did this work in the United States, so we could put these in the field in a place where they're not growing tobacco. And when we did that, what we found was that the plants continue to be about 30% larger even in the field, which is really important because it allows us to introduce a lot more variation that our crops are normally seeing to make sure that these pathways might be robust. But then, like I said to you, we know that at elevated temperature, rubisco actually makes this mistake far more often. So it fixes more oxygen, it's getting worse and worse. And so we grew them even at elevated temperature. You can see a bit of our technique here. We put these infrared heaters out in the field. We compared our little plots side by side to get them 5 degrees warmer. And what we found was that the plants even are able to put on more biomass under elevated temperature, and that they don't lose quite as much yield when you grow them at higher temperatures. And so they suffer less yield loss, which for plant researchers is truly one of our solutions that we're really hoping to find. Okay? And so the question we always get, is this really going to help? How is improving tobacco going to improve crops? I don't eat tobacco, and we shouldn't be working with the tobacco industries. And we don't. None of this work is funded by the tobacco industries because it's just our model. And so we have some promising work from other research groups looking at a similar pathway in rice grain, and we see a yield increase of about 27%. And my collaborators at the University of Illinois are also doing ongoing potato and soybean work to try to see, because rubisco is so universal, this one process happens in all leaves, if we look at other crops, do we see a similar benefit? Okay, but like I said, we're not only interested in biotech approaches. We don't even know if biotech approaches will ever be a culturally approved solution. So you cannot put all of your eggs in that basket, can you? Especially if what you're actually looking for are solutions. And so we have other sort of variation-based approaches to do it. So now we've shown we can rescue 30% biomass losses in this plant if we modify this process. We have an increase at elevated temperature in this process, or by manipulating this process. So now you can start to say, well, with yield gains that big, should we start looking at some natural variation? Maybe we won't see 30%, maybe we'll see 10, but that would be really good. And so we have a project looking at this in brassica at the moment. Current warming trajectories predict yield losses of about 30% in brassicas, and so can variation in photorespiration be exploited to mitigate that? And I think I end that here because I think it really shows this balance in plant biology at the moment of looking at plant biotech advances to then inform what we can do with conventional breeding approaches, right? So once you find that target, once you find variation in what you're looking for, you can start to then look for natural variation in populations for that. And so again, just a big thank you to some of the people who helped me do this work, both in my former group at the University of Illinois and now in our lab here at Essex. And I think we are happy for questions. So I am certainly happy to take any questions if you still have some for Tracy. I'm sure she used to. And if you're standing out in the rain and you want a seat, there are seats in here.
Yeah, thank you very much,
Thanks.
guys. We've still got a good 5-10 minutes if anyone has any questions at all. Yeah.
Yeah, hi. The genetic engineering and various other methods to increase crop yields, and we understand why, perhaps. But much of the information we're fed, probably by the press and so on, is that we waste 50% of our food.
Yeah.
And secondly, there is enough food in the world to feed everybody, but it's the distribution that's rubbish, and greed and so on.
Yep.
Perhaps the effort might be put in that direction as much as the rest, or perhaps some of your people are, I don't know.
Yeah, so I guess I have a twofold answer to that. Absolutely, I think we waste too much food and there is a distribution problem. But what we know as plant scientists is that we can actually go back through yield data over the last 20 years in regions that have been impacted by climate extremes. And so climate change is already impacting our ability to grow food. And we know that that pace is going to increase even more in the next 20 years. And so in regions particularly near the equator, what we're finding is that they're about 5 years away from climate catastrophes on their food supply, and that they've already been impacted. So rice, wheat, and maize yields are already down from where they should be from the breeding efforts because of changes in their growing conditions. So I don't doubt that we're wasting food, that there is food, you're absolutely right. That's a huge issue. And I think that's an issue that since I've been in the UK, I think this country's making real moves on, right? It's a topic of conversation here. This is not a question that you would get asked in Canada. We don't think that way yet, right? So it's very much part of the conversation here, which means that we are starting to make individual choices that would help. But in terms of plant science research, the most pressing issues that we have to start considering are what's going on with climate change. And I don't think that we can redistribute food to solve that. And I think we're seeing that right now with the looming food crisis from Russia's invasion of Ukraine, right? So wheat yields are going to be down significantly this year. And in several African countries, they're already feeling the pinch of that. And so Western countries, northern Western countries, are not giving wheat to those countries. So yeah, you're right, we should address that, but we're not. Tracy?
Can I just say something else about that, which I think you probably forgot a little bit? Is one of the projects that we both work on actually, that we're looking at some of these technologies, is actually to transfer some of that into some of the crops for sub-Saharan Africa. So cowpea, cassava, things like that. So although we're looking at those for some of our crops, for the crops we grow, we also have a big project that's trying to put that into cassava for sub-Saharan Africa through what we call the RIPE project. And so that's really trying to make those differences in the countries that require that increase in food now. So two-pronged approach, trying to increase and make our plants more sustainable that we're using, but also there's a huge program to put that into sub-Saharan Africa, where it's really going to be needed, and probably feeling those pinches with political and climate changes much sooner than we are.
Thanks, Tracy, that's a good point.
Sorry, I think I stopped someone else asking a question.
Sorry, have we got any more questions at all? Yeah?
Am I right in thinking that this is the difference between C4 and C3?
Yeah, that's exactly it. So all C3 plants have this problem.
Yeah. And so C4 plants, did they evolve in hotter places initially?
Yeah.
So will they not evolve in as the climate warms? And is maize not a C4 plant?
Yes. So maize is.
But maize is struggling. So
Yep.
I take it that it's getting hotter too quickly for evolution to work. But my problem with all this is that this is going to be done by companies. Companies are going to take out patents, and none of it will feed the poor.
So
mention it.
really, we've just mentioned that you're absolutely right on all of that. Yeah. So just the program that we've just been talking about is called the RIPE program. It's actually run by the Bill and Melinda Gates Foundation that Essex is a part of. It's run out of the University of Illinois, and we are partners in that program. They have actually set that up so that it cannot be taken over by companies. So it's they've actually put a program in place that we're working with at the minute that actually are doing that to make sure that the patents are not taken by the big companies, to make sure that they can go to sub-Saharan Africa, because that's another blocking point. We've seen that with a lot of golden rice and all of these things that we've heard about, and they're actually trying to do that to make sure that that technology is not taken up and swallowed into that sort of program and will go to sub-Saharan Africa as part of that program. It's called the RIPE program. RIPE, R-I-P-E, RIPE. Sorry, accent.
That's all right.
RIPE, yeah. So you look up RIPE. It's a Bill and Melinda Gates funded project. Been going for 10 years now, funded for quite a lot of money, very sort of generous funding as well. And we're actually looking to go for the next stage of that at the moment. That's made some of the research that Amanda showed us is under RIPE, and it's made some step changes in some really, really good science as well. That's beneficial for all of it, but targeting key crops for sub-Saharan Africa.
I believe that's all we've got time for from Tracy and Amanda, but a massive thank you. I think that was actually very interesting.